And I love the reframe of the hallmarks of aging being a symptom and really your nervous system balance being that upstream thing that you can have control over a lot more than you would think that would eventually downstream change the expression of those hallmarks.
You can do a whole bunch of things like vagus nerve stimulation to downregulate inflammation, or those taking deep breaths or other ways to activate the parasympathetic. You can take NMN or NR to help restore your NAD plus levels. I mean, simple intermittent fasting is all you need to restore the balance of AMPK and mTORC1. But when they're on permanently, when they're on chronically, they lead to some problems. One of which, and it's really important, is epigenetic drift. This is the slow change in how your DNA is expressed. Now, just to dive into the details of that for a second.
But before we get there, JP, I went into Google and I asked, how many theories of aging are there? There's well over 300. Why do you think we needed another one?
I have a quick favor to ask before we start. I'm not even asking 30 seconds of your time. If you have been enjoying the 90% Healthy podcast, which truly appreciate if you hit subscribe, it genuinely helps us grow and reach more people and keep bringing you thoughtful conversations about health that actually moves the needle without losing your mind. We're really, really grateful that you're here and continue to be. Now let's jump in the episode. Welcome back to the 90% Healthy podcast. I'm your co-host, Claudia, longevity journalist, coming to you from Naples, Florida, and I've got Don on the other hot seat.
Claudia, it has been a minute. You have been out gallivanting around the world. You've got to spend some time in Europe, and then we saw each other for the first time since we started the podcast. We were down in Austin, Texas at Beyond Biohacking, which was really exciting.
Yeah, it's really the community, and it was so nice to finally get to hang out and see you beyond the screen.
Yeah, there, no doubt. And what was, I was telling someone the thing that felt so good about getting down to Austin is the love that I felt from our community. I've been away from that community for a year and a half, almost 2 years. And God, it was great getting down there and seeing everybody.
Yeah, it is truly a high that you come out of when you go there. It's one of those loving connections that we all miss when we're working remotely. Yeah, but we have a guest today, and we have a very exciting guest today that, Don, you brought him on and brought him to my attention, and I read the paper that we're going to talk a little bit about. And before we introduce him to you guys, I wanted to kind of frame this as something we've talked about. We've talked about the nervous system, we've talked about aging, but we haven't talked about the two-in-one episode, because turns out there is a connection in how your nervous system is regulated, how balanced your sympathetic and parasympathetic is in relation to the hallmarks of aging. Now, turns out that those may be symptoms. So we have JP Errico here with us. Thank you, JP, for joining. And I'm excited to unpack this because there's a lot.
Yeah, there is.
So
it's just beginning.
Yeah, JP, you're an MIT grad. Did you study engineering there?
I did. I was an aeronautical engineer.
Okay. And then how did you wind up designing vagal nerve stimulators?
That's a long and very winding road.
Okay.
So I graduated from MIT and went down to Duke, really for law school, believe it or not. Went and started law school, became very, very dissatisfied with the loss of everything engineering and science related in my life, because that's certainly not something that they talk about over in the law school. Sure. So I marched myself over to the engineering school and said, hey, can I take some engineering classes at the same time? And ended up getting a master's degree in engineering while I was doing law school. Ended up focusing on patent law, which is sort of this is the one place where law and engineering sort of meet up. And then shortly thereafter, I was approached by my uncle, who is a world-renowned spine surgeon, to help him develop the next generation of spine products. He had some ideas, and when we sat down with each other, it became pretty apparent that both of us were very inventive, and I just needed to understand the mechanics of what was going on in spine to start to think of ways that we could stabilize segments and help to keep the motion the way it's supposed to and not become dysfunctional. And we ended up with a very successful business doing that. And it was sort of a return to what I grew up with in my house, because my father was an OBGYN also. So
Okay.
both brothers had been doctors. And so I worked with my uncle and I knew my dad, and the two of them sort of infused in me by osmosis, I guess, a lot of understanding around health and the practice of medicine. But I'm a scientist, so I started going back and taking classes in everything from organic chemistry and biochemistry and genetics, and enrolled myself in a PhD program in immunology, and realized that I kind of could just read the material myself, and then got involved in neuromodulation. And I'm sure we'll talk a little bit about that, but that's been a great stepping stone for me to study
Mhm.
everything about how the nervous system and the immune system are really two sides of the same coin. I actually refer to the autonomic nervous system is we have the innate immune system, we have the adaptive immune system, and then we have the proactive immune system. If you think
Mm,
about it, the
love that.
innate immune system
This is
is
great.
how we deal with outside influences that hit us. And it's
Mhm.
not always, chemical or physical. It can be emotional or stress. Those things are transduced into our biology through the autonomic nervous system. It's also transduced through the innate immune system, and then we learn about it. And so the next time we encounter something, we have an adaptive response. But, we don't want to have to go around drinking green water all the time. We had to develop a brain that recognized danger and said, we don't have to physically interact with it in order to avoid it. We can recognize it and avoid it. And that's our threat response. And that's our brain functioning the way it's supposed to.
Great explanation of the autonomic system. Now, we're here today to talk about a paper that you authored on the autonomic nervous system. But before we get there, JP, I went into Google and I asked, how many theories of aging are there? There's well over 300. Why do you think we needed another one?
Well, I think it's been a slow development over time. The first real development of ideas that were scientific and not, mythological sort of began in the late 1800s.
Okay.
And those evolved, and we had some crazy theories like the number of heartbeats. Everybody
Yep.
had a certain number of heartbeats you were given at birth, and once you used them up, you died. And you can go back. I mean, there's some crazy ideas. I mean, go back,
That's
and
why.
Aristotle believed that the brain was a radiator, and that all of the important stuff happened in your heart, and all the brain really did was radiate heat away. Obviously, that wasn't right. He was pretty good on a whole bunch of other things, but
Right,
that one
right.
he missed. But through the years, there have been theories that range from the really important ones are things like mitochondrial theory of aging, the telomere theory of aging, inflammaging. But the one I really, really got interested in and believe it's not even really considered a theory of aging. It's the cell danger response that Navio developed back. I think that paper came out in 2013 or 2014, where he talked about how cells respond to danger. And when I read about that, it started me thinking,
Hmm.
really just almost subconsciously, about how we perceive danger and how our bodies respond to danger is through the autonomic nervous system. And so my interest in aging got me thinking about and reading about how do we quantify this aging process? How do we understand the mechanisms that are driving it? And a lot of that drove me in the direction of reading the papers by Lopez Oten and his crew of collaborators on the hallmarks of aging. And we can go through what they are, but I bucket them into four
Yeah.
categories. There's there
That's a
Yeah.
brutal paper to read.
Yeah.
Okay.
It's
There's just no two ways about it. It's
yeah.
Well,
brutal.
there's two of them.
Yeah, yeah.
There's the one in 2013 and there's the one
Right.
in 2023 where they expanded. They started out with 9
Right.
and they
Went to
expanded it to 12. I looked at, look at the 12 hallmarks of aging, and I sort of bucket them into 4 categories. And they mostly fit into these categories. It was a
Mhm.
first shot at looking at things. You have the information bundle, and that includes, telomeres, telomere attrition, shortening of those telomeres. We can talk about how that happens. We have epigenetic drift, the changes in the control systems around our DNA, and then DNA mutations themselves. And then you have the second bucket, which is what I'll call the energy bucket. And that's mitochondrial dysfunction. obviously front and center.
Huge.
And then
Yep.
you've got stem cell exhaustion and you've got cellular senescence, which, they kind of fit in that bucket. And then I had two other buckets, which were the homeostatic buckets, the intracellular homeostasis, things like whether or not I'm making the right proteins and proteostasis, and whether or not I'm recognizing whether I should be in a catabolic state or in an anabolic state. And then on the other side, you've got the extracellular homeostasis, which is your inflammation. Are you communicating across cells correctly? Things like that. And then dysbiosis sort of fit, but it didn't fit. So I wasn't really sure where to put it, but it was extracellular. But the common theme that went through all of it was these are symptoms. These aren't mechanisms. They're symptoms of what happens when you age. We need to understand why they happen. And the first stop in there was to say, well, something has to be coordinating it, because as Elon Musk said, my right arm doesn't turn 70 years old while my left arm is, 20, so we have the whole system that we live in is aging at the same pace. Now, what the consequences of that aging might be different in my liver versus my brain versus my digestive tract, but that's not really a function of the processes themselves. It's a function of the susceptibility that tissue has.
Yeah. What's interesting about that, so what brought the three of us together is the concept of heart rate variability. I met Claudia years ago when we were out west. She was working with a company that measured HRV. At the time, I was moving into longevity and had just done a lot of work at the university level. You've obviously got an extensive background here, so that's what brought us all together. And on the podcast, we talk about it a lot. I'm sure our audience is getting an earful on this. But what you did, which really changed the way I think about some of these things, is you're looking at we treat the autonomic nervous system, or HRV, much like a thermostat and a thermometer. Things turn on and off, and we're constantly working the thermometer and thermostat based on practices that are influencing our sympathetic and our parasympathetic system. You're looking at this as, okay, every time that thermostat kicks on, your furnace is wearing out. And the more you kick the furnace on, and so this is when we're starting to go down this symptoms route in the hallmarks of aging. And I thought that was just a wonderful way to look at this, because being able to, it's, you did a great job of connecting HRV to the hallmarks of aging. So thank you for that. So as we start to take a look at this, one of the things that I pulled out of this, and you spent a lot of time talking about sympathetic activation.
Mm-hmm.
Our sympathetic system, I've said on the podcast 1,000 times, if you're born in a rattlesnake pit, the only way that you survive is through hypervigilance. Unfortunately, we don't live in rattlesnake pits, but we have paper rattlesnakes.
Very fortunate that we don't. Fortunate that we don't.
Yeah, but we have paper rattlesnakes around us all the time. In your opinion, as you look at this, that sympathetic stimulus, is it enough to recognize the stimulus and then react to it? Or do we need to get to where we don't live in a rattlesnake pit, or we don't even recognize paper rattlesnakes anymore?
So I think it's a combination of both. I think there's no question that recognizing stress and reframing it, for example, reframing it as an opportunity to learn.
Okay.
An opportunity to change. Growth is not easy. When was the last time you grew as a person as a result of something that was simple and didn't stress you?
No.
Nope, that does not work.
Well, that doesn't work. So we have to recognize that some stresses are really valuable, and we actually refer to them as hormetic stresses. In fact, my high school, I went to a Catholic high school, and our motto was in Latin. It was successive arrescit, which means that which gets cut down grows back stronger. I mean, I grew up with this. I mean, this is the whole idea of, literally hormetic stress makes you stronger. That whole, Nietzsche line, that which does not kill us makes us stronger. The truth is, if it beats you up long enough, you're going to end up in therapy forever.
Right.
But I think there's truth to that. But at the same time, we have, independent of your politics, I'm not talking about a specific president, but the general rule has been that the president of the United States has a 4-year term during which he ages 10 years.
Dramatically,
And it
yes.
is not because he has a lack of medical care. I mean, he's got the best medical care humans could possibly muster.
Right.
And yet the chronic stress of that position leads to an acceleration of aging. And that one thought, I remember I was out on a run when I was thinking about that. And I was thinking to myself, that's such a great analogy. Oh, wait a minute. That tells us what is the transducer. That tells us that the autonomic nervous system is transducing all of that stress, all of those things. And it doesn't have to be physical stress. It doesn't have to be mental stress. It can be emotional stress. It can be anything. It can be toxins, ionizing radiation, you name it. Those are the external factors that we have to interpret. You know, we talked about how the innate immune system, you know, has to come in contact with something. And the same thing is true with the adaptive immune system. But the proactive immune system, that's the brain, that can just see it and be engulfed in it and will activate the immune system. It will activate the cell danger response, which brings us back to Navio. But the question is, how does that happen? How does the cell danger response, the cascade of different things that happen at the cellular level, how does that happen from, you know, as I say, you can be chased by a lion, or you can be chased by a deadline, or you can be chased by an angry spouse. You know, any one of those things is going to trigger an immune response. It's going to trigger a heightened level of reactive oxygen species. It's going to help deplete your NAD+ levels. It's going to dysregulate your catabolic anabolic balance. All of that. And those are the cell danger responses. And we had to figure out, how does that happen? And it turns out that the autonomic nervous system and its mirror image, which is the innate immune system, do that. They do it for us, you know, in a negative way, because every time we have our threat response mode on, we're going to kick that into gear. But that's why you can have two guys in, you know, the same Humvee and went to the same high school, and they're in the military together, and one comes out of the military and he's got problems, and the other one didn't. He's fine. He becomes a bank president. He marries his high school sweetheart, and has a wonderful life. It's how we perceive the stress that matters.
But then also, how do we handle the stress response? Because I think if we talk about those two individuals in the military, yes, they may perceive the same situation in a different way, but also when we are all exposed to the same stressor, people have different tools to adapt, different tools to respond. How does that change our cellular environment based on how we respond to that stressor?
Well, two things. One is, and we talk about this a lot, is you have to get back into what I refer to as parasympathetic recovery mode. So you have to be able to get yourself back out of that stress state into a parasympathetic state. You just need to relax. I mean, we have
easier said than done for some people.
No, you're absolutely right. And some people, I mean, it's in our language. We tell a friend who's upset about something, hey, you need to chill out.
Yeah.
Or we say, hey, you need to take a deep breath, or go take a walk. I mean, we have it in our language and in our expressions because of the truth of it. And so, yes, when you want to get yourself out of those situations where you're chronically stressed, the best thing to do is to break it up by doing exactly what we just talked about. Take a deep breath, go for a walk, chill out, relax for 5 minutes. I talk about, frequently when I talk to people about how to do exactly what we're talking about now, I talk about the Galloway technique that I learned when I was training for a marathon. Back in 2000, I ran the New York Marathon, and I had a training partner, and we would go out and we would do all the training associated with the New York Road Runners Club schedule for, getting to the point where you could run a marathon. And the way that program works is you go out, and on Tuesdays and Wednesdays and Thursdays, you run short distances. But every Saturday, you go out and you run a really long run. And it starts out being 6 miles, and then it's 8 miles, and then it's 10 miles, and then it's 13 miles. And we were doing fine. We were going along great. We did the 10-mile run. It was awesome. We did the 13-mile run. It was terrible. I mean, it was awful. The last 3 miles, we completely bonked to the point where it took us 45 minutes to do that last 3 miles. And I thought to myself, oh my God, that's only halfway. Well, we're going to start this run on November 4th, and we're not going to end till like the 8th. I mean, at that pace. And so what I did was I said, we got to come up with a radical plan. We got to figure out something to do. And there was a guy named, his last name is Galloway. I can't remember his first name. And he trains people for marathons, and he has a technique, which is you run for 9 minutes and then you walk for 1 minute. Sound familiar to what we're talking about getting a little break?
Yeah.
He says, take a 9-minute run, 1 minute walk. You're going to lose some time with that 1 minute walk the first time you do it. But by the second or third time you do that, what you gain in that rest will make you so much faster. So I went out and I said, I'm going to do that for the 16-mile run, which was the next run. And I thought to myself, let's see whether it works. And I finished the 16-mile run faster than I had run the 13-mile run.
Impressive.
And I felt at the end of that 13-mile run, I said, there's no way I could do another 13. When I finished the 16-mile run, I thought to myself, I could go another 10. Doing this, I could do it. And so the rest of the training was easy. I mean, we got to the marathon and it was easy. I mean, just run for 9 minutes and walk for 1 minute. Use that same strategy in your life. I say to people, if you got an office job, if there's stress, from 9 o'clock till 9:55, work really hard. Focus, work really hard. But for the last 5 minutes of that hour, put the phone down, push yourself away from the desk, and do like 4 or 5 things, okay? 1, stretch. 2, take a few deep breaths. 3, get up and go to the bathroom. We're not in kindergarten. You don't have to raise your hand. Go to the bathroom. Get a drink of water, because not going to the bathroom and not being hydrated are 2 really great ways to activate your sympathetic nervous system, okay? And then last, make a plan with somebody, either a coworker or a friend, or or call your spouse and say, hey, let's do something this weekend. I was thinking about something fun. What do you think? And then get back to work at the top of the hour. Listen, it's what we used to call a smoking break.
That's
Okay.
true.
That used to work. Okay. Now, maybe smoking wasn't the best thing to be doing during that time. Maybe it was stretching or doing some deep knee bends or whatever, but do something to, hit that easy button. Okay. Take a moment and relax, because it gets you back into that parasympathetic recovery mode. And I'll tell you, when I tell people this, they come back to me and they say, before I did that, when I got out of work at 5 o'clock, I was shot. I had nothing left for my family. I had nothing left for my kids. And now I'm energetic. I'm home. I'm in a good mood. I cook dinner now. I have time for my kids and their homework. And I think to myself, yeah. And all it was a couple of 5-minute breaks. So, that's the best way to break it up. And some people do it naturally and some people don't. And for the people who don't, learn some simple techniques for doing it, like the Galloway technique I talk about here.
Yeah.
And it'll be life-changing.
Because a lot of people get stuck in the hamster wheel, just going, and they think more is more. But I think a lot of athletes, and I'm sure Don can speak to this, understand the value of the recovery, because that's where adaptation happens. If you give time to that parasympathetic boost, then you can push further.
It's the power of pause. Well, there was a whole ad campaign around that. I can't remember if it was Gatorade or Adidas or somebody, but they had a whole bunch of athletes talking about the power of pause. It's the power of taking some time off so that your body can heal, but it's also your psychology can heal, your resilience overall can heal. It's at the immune system level and at the autonomic nervous system level.
I wanted to ask, just to kind of get back on track in terms of the aging and the adaptation process. So we've talked about how powerful adaptation is. When you have that pause, you can push further. Now, what happens to that aging process with maladaptation when we are constantly pushing into that chronic sympathetic state?
So it's going to get a little technical here.
Please, we love technical stuff.
Good. So we've talked about now the sort of the external influences, which I call layer 1 in my model. Okay. And then it's all transduced, or mostly transduced, through the neuroimmune axis, this threat response mode. Okay. When it's short term, that's fine. Then you get to resolution. The cell danger response has 3 stages to it. Only the first one is inflammatory. The rest is resolving. Okay. But if you never get to the resolving because you're constantly pushing the gas pedal on the cell danger response, then you end up with a chronic state. And there's really 4 big cell danger response pieces that we just talked about: mitochondrial dysfunction, NAD plus depletion, chronic NF kappa B activation, which is your inflammation, and then this imbalance between AMPK and mTORC1, which we can get into the details of that. But it's basically telling your cells whether they should be in growth mode or in, spring cleaning recycle mode. And you need to get into that spring cleaning recycle mode frequently. But once you have those, they're all reversible. All of those things are reversible. You can take urolithin A and methylene blue, and you can take MOTS-C if you want to inject it, and you can do a whole bunch of different things to help your mitochondria. You can do a whole bunch of things like vagus nerve stimulation to downregulate inflammation, or those taking deep breaths, or other ways to activate the parasympathetic. You can take NMN or NR to help restore your NAD plus levels. I mean, simple intermittent fasting is all you need to restore the balance of AMPK and mTORC1. But when they're on permanently, when they're on chronically, they lead to some problems. One of which, and it's really important, is epigenetic drift. This is the slow change in how your DNA is expressed. Now, just to dive into the details of that for a second, there's multiple different ways in which we control which genes get expressed in which cells. Because a skin cell shouldn't be making the proteins that a liver cell makes, and a brain cell doesn't need half the proteins that are made in your pancreas. Okay, so what you have is a set of controls which control which genes get expressed. And one way that happens is through DNA methylation. So it's a chemical modification that takes place at what are called CPG islands. It's the cytosines in your DNA being methylated. Okay. Another way is to modify the proteins, the histones that the DNA is wrapped around. So, should it be tightly wrapped or should it be loosely wrapped? That's something we call histone modifications. There's a third piece of this equation, which is, non-coding microRNAs, but we don't have to get into that right now. What we have is in epigenetic drift, this slow deregulation or slow loss of access to the right proteins and a gain of the wrong proteins being manufactured in the cell. And it's a fascinating way that this drift, which you think is kind of random, actually turns out not to be random. It actually drives a chronically increasing level of inflammation. And the way that happens is every gene has a promoter sequence, or I shouldn't say every, but almost every gene has a promoter sequence. It's a sequence of the DNA that tells the machinery that's going to copy that gene where to attach and where to start. Now, if that's not exposed, then that gene's not going to be made. It turns out that the pro-longevity genes, things like FOXO3 and PGC1 alpha and things like that, those genes have promoter sequences that have a lot of those CPG islands. Okay, a lot of those CPGs. So when methylation happens, it tends to happen to stop the longevity genes from being made. On the opposite side of the spectrum, you have the inflammatory genes. The inflammatory genes have their own sets of promoters, and those promoters don't have many CPG islands. So when methylation occurs randomly, because it's not that well controlled, when it's randomly happening, it's non-randomly and in a biased way, it's suppressing longevity. It's slowly breaking down our ability to regenerate and heal and restore ourselves. And it's slowly increasing the level to which we're going to make inflammatory cytokines and we're going to get stuck in that inflammatory state. So that is one of the most important, if not the most important to me, steps in the aging process, because it's irreversible. Or up until recently, it's been considered irreversible. Everything we talked about first, the mitochondria, the NAD, I told you all the different ways that you can do those, you can reverse. Epigenetics is not so reversible. It's difficult.
What are your thoughts about the work that David Sinclair is doing right now up at Harvard with the Yakanama factors and the reprogramming at that level. Do you think that do you long term, do you think that's a pathway that's going to deliver benefits?
Yes, I actually think that that's a very, very interesting strategy. It started, as you said, by by Shinya Yamanaka, who was a doctor over in Japan back in 2006, 2007. He published a series of papers in which he showed how you could take an adult skin cell from, a 70-year-old person. You could take a skin cell and you could literally reverse the age of that cell all the way back, and the differentiation status all the way back up to being a stem cell, an induced pluripotent stem cell. I don't know if, your listeners may recall, but back in, the late 90s, there was a lot of controversy about the use of embryonic stem cells.
Yeah.
I mean, a tremendous amount of media time about the pro-life groups and the pro-choice groups being opposed to the use of stem cells. That's largely gone. And it certainly isn't because those two groups decided to hold hands and sing Kumbaya. It's largely because of the work of Shinya Yamanaka. What he showed was that you didn't need embryonic stem cells. We could make those stem cells from tissue from a 90-year-old. You could take a cell and you could reverse it. Now, that was really exciting, but you can't take, that technology and turn a human being from a 70-year-old to a 30-year-old, because you're just going to convert everybody back to a bunch of stem cells. And you actually need skin cells, and you need liver cells, and you need brain cells. You can't just all be, pluripotent stem cells. But a group about 8 years later, led by a guy by the name of Juan Carlos Espesua Belmonte, did a study in which he only turned on those genes, because there's 4 genes, O, S, K, and M. That's OCT4, SOX2, KLF4, and CMEC.
Okay.
If you turn them on for a brief period of time, that the process actually happens in 2 steps. And it didn't have to, which is really the fascinating part to me. It didn't have to. But what it does is it takes that old cell and it fully rejuvenates its epigenetic marks. It changes it all the way back to where it was when it first differentiated as a young cell. And that affects the mitochondria, it affects proteostasis, it affects everything. And so the cell literally becomes young again. And then it will, only after it's reached that youthful state, will it then go back and do the de-differentiation back up to a stem cell. So he was able to take animals. He did this in rodents. Okay. He took these animals that were aged and infirmed, and he was able to make them young again. Now, he did it through pretty elaborate means, but David Sinclair and others have looked and said, maybe we don't have to get involved in elaborate gene therapies and all of these things that would be very difficult to get through the FDA. Maybe we can do it with either small molecule drugs or in a way that's a lot more tolerable for humans. And in fact, there's a really cool project going on at a company called Retro Biosciences, which is being funded by Sam Altman of ChatGPT. I think he put $180 million into it. But what they're doing is they're using ChatGPTs, or I guess an LLM, a 4.0 model, I think, that they programmed up with millions of data points to show that they could rebuild OS and K, and not use M, but OS and K in a way that is different from anything that biology's ever produced before. So these are new, novel proteins, but they're like 50 to 100 times more effective. And so pretty exciting stuff is coming down the pike. I tell people, the person who is going to live to be 1,000 years old has already been born. And I think that, we're, I'm probably the oldest of the group, if not Don, Claudia, I think you're probably younger by a decade or 2 at least. But I think that my kids, and maybe I, will be able to live for a substantially longer period of time than my parents and grandparents will.
I mean, it's interesting you mentioned Sam Altman's company, because there was another one that just recently raised over $430 million new limit that is working on human cell reprogramming. And they're looking at, obviously, diseases of aging like alcohol-related liver disease to reverse the age of that liver cell, hence get rid of the existing disease. I mean, it's
Absolutely.
Coinbase CEO is Brian Armstrong's the company,
Mhm.
or he is one of the co-founders. So there's a lot of buzz
Put
in
100 he put 100
reversing.
million I think he put $100 million of his own money in,
Oh,
and they just
wow.
did 430. And listen,
Yeah.
Altos Labs was, their A round of financing was $3 billion. It was the largest A round, I think, at that time. And
Yeah.
and that's funded by Jeff Bezos and Peter Thiel and a bunch of other guys who are these tech billionaires are not stupid people.
Hmm.
I mean, Google has their own effort called Calico, the California Life Company.
Wow.
These
Yeah.
tech billionaires are very, very smart, and they have latched on to the idea that reversal of aging is no longer a subject of mythology. It is no
Hmm.
longer something that we are going to have to rely on Ponce de Leon to find the fountain of youth.
Yes. Yeah.
We have the scientific knowledge as to how this can be done. We're not there yet, but I think partial reprogramming, which is what this technology is called, is a very interesting approach. It's not the only approach. In fact, there's another approach that just came out of China last year, was published in Cell. I think the actual publication date was September, but it came out in July. And it is absolutely fascinating because it may ultimately work through a very similar process, but it's using human stem cells. And they did this study in monkeys, in macaques, and they were able to reverse the ages of these macaques using infusions of stem cells that were
Hmm.
human stem cells that were modified. And the animals had a rejuvenation, depending on the tissue they were looking at, of anywhere from 6 years to 25 years. And to me, the part of it that is most fascinating, because you could say, oh, well, okay, the skin looked younger. I mean, you could do that with creams. Their hair looked younger. Yeah, but so did their hippocampus. So did their
That
liver,
matters. Yeah.
okay? And the females that were already through and beyond breeding age
Hmm.
were able to become reproductively viable, get pregnant,
Impressive.
and
Wow.
bear offspring,
I mean,
viable offspring.
wow.
And that to me is you can't fake that. You're
Yeah.
not faking that.
I mean, imagine if women would just be able to go into menopause a lot later, how much their quality of life would improve.
Yeah, and and
Yeah.
and how about the pressure to have children?
Yeah.
Okay. How about the pressure to have
Yeah.
children before your career has been launched,
Right.
or getting to the point in your career where you're quote unquote powerful enough to take off
Yeah.
a year or two and come back and not have to start over again? I
Yeah.
mean, all of that is within reach, and it's super exciting. I mean, I thought that developing a non-invasive vagus nerve stimulator and all the things that vagus nerve stimulation could do would be the pinnacle, but it was just the stepping stone. It's just the beginning of what we're going to be able to do with things like partial reprogramming, things like FOXO3 enhanced stem cells, things like natural killer cell infusions, things like vagus nerve stimulation obviously is part of it. But these techniques that are being developed right now
Yeah.
are going to change the life expectancy of humans exponentially.
Yes. 100%. And it's so interesting to just watch this industry unfold at a speed, at a pace that it just hasn't been. Like it's insane. And a lot of the things that we've talked about just within the past 10 minutes obviously aren't necessarily accessible to the masses. But I want to kind of drill back into the nervous systems, things that everybody who's listening have access to right
Mhm.
now to modulate, to regulate in a way that could slow down their aging process. And I love the reframe of the hallmarks of aging being a symptom and really your nervous system balance being that upstream thing that you can have control over a lot more than you would think, that would eventually downstream change the expression of those hallmarks. How would you help our listeners and our audience take control of that? And then how would you help them explain to them in a way that would really make sense for them to, okay,
Mhm.
certain small steps and practices I can do on a daily basis that would really help me slow down my aging process and not have stress kill me down the road?
Yeah. So I think that's a great way it's a great question, and I'll frame it the following way. I actually just gave this analogy to my daughter. Imagine you wanted to stop the Mississippi River flowing through New Orleans, okay, or Louisiana. Be pretty difficult to do. I mean, at that point, there's trillions of gallons of water that are pouring through that river. You wouldn't want to build a dam there. Because where's the water going to go? You're going to need to find a better spot to stop the flow of that water. I personally would say, why not go back up to Minnesota? Go up to where the headwaters of the Mississippi are and block it off there. Okay. You're not going to stop it down in Louisiana. You're going to stop it up in Minneapolis or Minnesota. Yeah. Okay. So the way to stop it is to starting early in life. First off, control the layer one inputs, microplastics, toxins, don't make drinking alcohol, a regular thing that you do. Don't eat a lot of junk food, don't get addicted to ultra processed foods. Make certain that sleep becomes an important thing to you and that you prioritize it. Exercise. Nothing is worse than having a low VO2 max. Okay. For those of you who don't know, VO2 max is basically, I'm sure any listener to Don is going to know what VO2 max is.
Yes, we have talked about it, but it's always nice to reiterate.
Yeah. I mean, listen, it's really important. It's really, really important. But know that all of those things, those external things and those lifestyle choices that you're making have to be interpreted through the autonomic nervous system into whether or not it's going to trigger the cell danger response. And if it does trigger the cell danger response, how can you get yourself out of it relatively quickly and back into a state where the cell danger response can go through stage 2 and 3, which are the resolving stages, and not remain chronically in stage 1? Vagus nerve stimulation is a great way to do that. Anything that increases your heart rate variability is good for that. Things like deep breathing, exercise, cold plunges, you name it. Hormetic stresses will increase your resilience. But once you get to the point where some of that damage is already starting to happen, which, listen, by the time you're 25, it's already happening regularly.
For
it's probably
sure.
happening for
Most
some
people.
people.
Are starting too late.
Yeah. But the beautiful thing is, it's never too late to stop the aging process. And you can do that. We talked about vagus nerve stimulation and parasympathetic recovery mode. But then we talked about layer 1. And layer 2 of the model is mitochondrial dysfunction, and specifically electron transport chain leakage. Okay, so we all know how mitochondria work. We all heard about them being the power plant of the cell. You can function on glucose, or you can function on glutamine, or you can function on fatty acids. You want to function on fatty acids. You want to be in some level of ketosis, not ketosis like you've got diabetes, but but real functional therapeutic ketosis where you feel really good, really sharp. What you're going to slow down is insulin resistance. You're going to slow down endocrine dysregulation. Your hormones, your thyroid hormone, your sex hormones, those things are really critically important to maintain and to slow down that epigenetic drift. Now, we talked about how you're going to reverse that ultimately with partial reprogramming, but those things are really difficult to reverse at that stage. You got to do it earlier. And they then lead to the next layer of molecular problems, which there's 7 of them, and we don't have to go into detail about them, but that's where your telomere attrition comes in. That's where your ability to recycle your mitochondria with PGC1 alpha, your ability to trigger autophagy with FOXO3, your DNA damage response, which, yes, it's a really important set of signals and pathways, but when it's chronically active, let's dig into that for a second. There's different types of DNA damage. You can have a single strand break, or you can have the double strand break. Okay. There's 2 different systems that are going to try to heal that damage in the cell. And it happens millions and millions of times a day. Okay. And your cells are really good at fixing that DNA. It's the ATM and ATR responses. But they both work through something called P53. And I don't know if you've ever gotten into P53, but P53 is a pretty important enzyme. It's involved in helping to restore the DNA and not letting that cell become cancerous. And how does it do that? It does that by blocking the whole cell cycle. It slows it down because you can't be fixing the DNA while you're trying to copy it to reproduce.
Yeah.
Okay. So P53 is the trigger to arrest the cell cycle, slow it down. Okay. Give the DNA damage response proteins a chance to get in and fix things. Okay. Sort of like you can't drive your car while you take it to the shop to get fixed. Okay. You got to take it to the shop and leave. But
Yeah.
if the damage is constantly happening and it's chronically on, then the cells never restore and you end up in a senescent cell. You end up with cellular senescence. Those are the zombie cells that people talk about. That's how it happens. It happens because you've got this chronic damage being done to the DNA by reactive oxygen species that are coming out as a result of the cell danger response and the breakdown of the mitochondrial electron transport chain and the leakage that occurs. So now we've literally gone from that's where it starts all the way through to cellular senescence. Okay, cellular senescence is pretty far down the road.
It's
Towards
pretty
aging.
far down. Yeah.
Yeah.
And there are solutions nowadays that are trying to address those and just clear out senescent cells so you can
Yep.
at least have less garbage, have less zombie and trash inside lingering, creating these what is it called? These inflammatory markers and inflammatory cytokines that they
The SASP.
initially also produce.
Yes, that's the senescent-associated secretory phenotype
Yes.
that you're talking
Yeah,
about.
yes.
SASP.
Very
Yes,
interesting.
absolutely. There's lots of really great things that are coming out, and some of them are simple. You know, there's some supplements, things like quercetin, fisetin. You know, those are real easy things that
Yep.
you can just get from Amazon or from any health food store. And then there's some pretty elaborate drugs, things that are like chemotherapy drugs that are being used, like dasatinib.
Yep.
That's sometimes coupled with quercetin. But I'm a big personally, I'm a big believer in amplifying what your body does normally. And what your body does normally in order to clear out senescent cells is it sends out a group of cells called natural killer cells. And these are immune cells that are really good at clearing out cancer cells and senescent cells. They recognize the markers that are on the surface of these senescent cells, and it immediately it's like balloons popping. You know, these things, they just go around, they pop these senescent cells, and they digest them away, and they're gone. So it's pretty remarkable. The problem is the older you get, the fewer of these natural killer cells that you're producing. And here's a great way to enhance your natural killer cell levels just naturally: vagus nerve stimulation. There was a study that we did over in the UK with a great researcher, Wen-Fai Ng, and what they showed was that in patients with autoimmune diseases, because that's what we were studying, they did a lot of blood marker work, and what they showed was that vagus nerve stimulation rapidly and remarkably increased natural killer cell expression. We were actually a little worried about that because we were like,
Impressive.
well, it's actually supposed to calm down the immune system. But it did with things like that were inflammatory, but it upregulated natural killer cells. So if you want to reduce senescent cell burden and you don't want to necessarily go on a chemotherapy agent or wait for natural killer cell infusions of your own, because those are very expensive.
Yeah.
You can just get a vagus nerve stimulator.
That is so interesting. And of course, we've had your podcast co-host, Dr. Nawaz Habib, on our podcast before.
Mhm.
So we've talked plenty about vagus nerve, and both Don and I are a massive fan of that technology because it really just puts your body in a parasympathetic state through such a non-invasive way, and it doesn't require a lot of time.
It doesn't at all. It's 2 minutes in the morning and 2
Yeah.
minutes in the evening. It's so simple. I wish, I'm one of the inventors of the technology. I wish that I could say that I knew all of this was going to be the result. I didn't know it all. I had no clue. I had no clue about most things about this. It was really just me as an engineer reading one paper. I read one paper. There was a neurosurgeon that I was working on some neuromodulation stuff. And he said to me, anytime anybody has ever cut a nerve and as a result of cutting the nerve gained a benefit, you might be able to stimulate the nerve and save it and still get the same benefit. And so I said, okay, put that in the back of my head, waited about 4 years and sat one Saturday afternoon looking on the internet to try to find instances where somebody had cut a nerve and gained a benefit. And one of the articles that I read was out of the old Soviet Union. They had taken dogs, that were allergic to, they made them allergic to egg white, and they exposed them to what would be a lethal anaphylactic reaction, except they cut the vagus nerve in half the dogs, and the dogs that had their vagus nerves cut all
Wow.
survived. Yeah, and
Wow, that's a
unfortunately,
wild experiment.
no, it was a while. You probably couldn't do that anywhere else other than the Soviet Union. It was like,
Oh my
but
goodness.
the problem was they didn't take it any further. And I was sitting there thinking to myself, I guess they, in their Russian way, they said, "We now have cure for anaphylaxis. We just cut your vagus nerve." But but we went to Columbia University and duplicated the research, not in dogs but in guinea pigs, and and rabbits, and we saved them. We could, with
Wow.
vagus nerve stimulation, we could take an animal that was programmed to have a lethal anaphylactic reaction, and we could save them. And that's where it
Why
started. Yeah.
why do you think vagus nerve has such a massive power over our livelihood?
So I think it starts with the miraculous work that was done by Kevin Tracy back in 2000 with his team at the Feinstein Institute. I give him all the credit. He scooped me by 5 years. I truly didn't have a clue about any of the work that he had done when we started, but what we did in anaphylactic shock and ultimately in hypovolemic shock and in once we read his work, we went back and did it in septic shock as well. But he started out in sepsis looking at a protein. It was a polypeptide called cemipimod. At the time it was just a number. It was CNI 1341, I think, or 1391 or something like that. And he had injected it into animals who were going through septic shock, and he showed that it could help with sepsis. It could block the lethal process. And he tried to figure out, well, how's it working? And he's a neurosurgeon by training, so I guess it was just interesting to him to inject it into the brain. And when he did, in tiny, minute quantities, it still had the same benefit.
Huh.
And so what he did was he looked and he said, well, where's that working? And it was working at the dorsal motor nucleus of the vagus. So
Mm.
basically the root of the vagus in the brain. And what he discovered was what's now referred to as the cholinergic anti-inflammatory pathway. That when you activate the vagus nerve, when you activate that parasympathetic system, it's doing exactly what you want, which is helping to resolve the cell danger response. It's getting you out of that cell danger response stage 1 into the second and third stages, which is resolving it and getting it back to homeostasis. And so it pushes you away from being inflamed into the direction of being quiescent. And that's meaning your immune cells. And so it has this profound effect. He injected animals with LPS. This is the study that actually ended up in Nature.
Mhm.
He took animals, rats, and he measured their innate levels of the cytokine TNF alpha, which is, the archetypal pro-inflammatory cytokine. He measured it, and it was, relatively low, almost immeasurably low. And then he injected LPS, which is lipopolysaccharide, and it's mimicking basically a severe bacterial infection. And the cytokine levels shot through the roof. They were massive. And then he cut the vagus nerve. And in his model, it actually got worse, which is interesting because it was different in what we saw in the animals that were in the Soviet study. And then he stimulated the cut end of the vagus nerve. And what he found was that when he stimulated that, he could drop the cytokine levels by 90%. So it was pretty remarkable work
That
that he
is
did.
remarkable.
He did a lot of work in that. He's done a lot of work in that. I think if there's a Nobel Prize handed out for this, he's first in line. What we did was 5 years later. But I kind of think of it as the difference between Bernoulli, who figured out how, airfoils worked,
Mhm.
and then you got the Wright brothers who actually invented the airplane. He's Bernoulli. Okay. He's, legendary for having come up with this model. We just figured out how to make the airplane. And, I use that same analogy. I say,
No, that's
while everybody else was flying around in biplanes, we decided to make the F-16. And we did it with a non-invasive handheld device that you can use for 2 minutes in the morning and 2 minutes in the evening. There's no surgery involved, no implantation of something into your body. And it has these remarkable benefits.
and of course, those benefits are shown pretty clearly in somebody's heart rate variability. And I'm sure you've seen some of the
Yes.
data. And I know we're going to come up to the end of this conversation shortly, but I just want our audience to walk away with knowing that doing vagus nerve stimulation is something that can be validated through heart rate variability, which a lot
Yes.
of our listeners are tracking. How would it show up in what? I mean, obviously, we know everybody's different, but in your research, what have you seen in terms of HRV's response to vagus nerve stimulation?
So I'm sure your audience is very familiar with what HRV is. There is a derivative marker called cardiac vagal tone,
Mhm.
which is basically the high frequency components of heart rate variability. And what we did was we did a study in Denmark with a group of people in Copenhagen. And what they did was they took 20 volunteers and they used the vagus nerve stimulator on them one time. So for 2 minutes, and that was it. And what they found in all 20 volunteers, every single one of them had within 90 seconds an increase in heart rate variability in the cardiac vagal tone that peaked out in the measurements at 90 minutes, and it was still elevated at 24 hours, statistically significantly. So it worked in everyone. So what we generally see is, and cardiac vagal tone is a quicker response time than heart rate variability overall. But what we have seen virtually across the board is that if you use it once or twice a day, you're going to see an increase in your heart rate variability that could be as little, and when I say little, you're going to say, well, that's a lot, 50% improvement.
Mhm.
So, take you from 30 to 45.
Yeah.
But I also have seen people who a year out from using it, their heart rate variability went from 30 to 120.
Impressive.
So, I mean, literally a 4x improvement.
That's wild.
Now, is that still going to remain high through something like a car accident or a surgery or getting drunk? No. Obviously, those things are going to suppress your heart rate variability, but your return to that higher level is going to happen more rapidly, and you won't dip down as far.
Which is how I describe resilience, what you're building with this tool. You're building your resilience and your ability to bounce back when life hits you, which inevitably will. And that's why we love talking about here on the 90% Healthy podcast. We want you to raise your resilience and your resilience score to a point where you're not taken out by a virus. You're not taken out by something small that would literally wreck other people. And I think raising your resilience through through the nervous system approach can really help your body put up a little bit of a stronger fight when
Yeah.
something else comes along the way. Yeah.
Yeah, absolutely. I'm sure you guys have talked about Wim Hof and his remarkable use of the parasympathetic nervous system to literally, he's taken on, what would be lethal doses
Yeah.
of LPS, and just through regulated breathing,
Hmm.
he's able to survive it and not even have, significant symptoms.
I mean, I
So
wonder if it has to do with the increase of natural killer cells that would potentially be produced with that practice
yeah,
that
no, I
help.
think that would be part of it, but I think that in his case, the LPS that he's taken on is driving release of cytokines,
Hmm.
but he can suppress the release of cytokines through the
Oh, yes.
cholinergic anti-inflammatory pathway. So
Cool.
it's
Yeah.
remarkable stuff. So
Very
it's
interesting.
exciting.
Okay. Is there anything we haven't talked about that you have coming up, a research, a book, a study, something that you want our listeners to keep an eye out?
Gosh, yes. I'll tell you a couple of things. One is, and we've published a couple of papers so far, but the final data is coming out shortly.
Hmm.
I sponsor some work up at the University of Victoria in British Columbia with a phenomenal colleague, a phenomenal researcher by the name of Marie-Eve Tremblay. And she and her team have done a lot of work looking at maternal immune activation as a trigger for autism and for schizophrenia and for a number of other neurodevelopmental conditions. And she was on my podcast and I was asking her about it. And I said, I think that vagus nerve stimulation used by mothers during pregnancy could reduce the risk of autism. And I would like to study that in your animal models. So it took a couple of years, but we did it. And the results look really good.
Wow.
So if women who are pregnant are worried that there's that an immune response that they might have could lead to autism. And it's like 1 in 12 boys now in California. I would suggest using vagus nerve stimulation because it can reduce the damage that's done in utero, not intentionally, but it happens because we're so flooded with immune activators that it's almost impossible to avoid them. And if you're susceptible, that's something that can happen. So that's one thing that works coming out. In fact, I think she just spoke about it at the Nobel conference, which
Wow.
is kind of amazing. I think
Yeah.
that's what she spoke about there.
Yeah.
And the other thing is we're looking at, there's going to be a series over the next 18 months of about 20 papers that we have coming out with collaborators all over the world. And they are taking all of the age-related diseases. So everything from neurodegeneration and atherosclerosis to hearing loss and cancer, and looking at how they are actually tissue-specific manifestations of the aging process. And that when you try to treat those diseases only one layer up, and that's layer 6. If you're only going to like layer 4 or layer 5, you're playing whack-a-mole. I mean, you're not going to solve the problem because the
100%.
underlying problems that are driving it are all the way back. It's like trying to build the dam in Louisiana. You're not going to stop the water. Okay.
Yeah.
You're really not treating the disease. You're treating a symptom at that level. You need to get upstream and start treating it at layer 1, layer 2, layer 3. That's where you're going to get the biggest bang for the buck and where things are reversible. So I think, we're going to be exposing some of the things like, is LDL really the thing that we should be focusing on in
Yeah.
atherosclerosis? Is beta amyloid really the problem in Alzheimer's? Is kidney disease actually more like atherosclerosis than it's like anything else? We're going to show people that.
Gosh, those questions. I can't wait to hear the answers. We're going to have you back on once those results and once everything is officially out and you can talk about it, because I think this is very interesting in the context of what our listeners are curious to know. And by then, I'm sure Don will have the internet all ready to go for some. He had a power outage. So unfortunately, we lost him about a few, 10, 15 minutes in. But it's okay. I'm sure, JP, we're going to have you back on. But in the meantime, if somebody wants to find you and read your stuff, where can they go and find more about you?
I'm actually kind of not tied into. Obviously, the Health Upgrade podcast is my biggest foray out there. I'm kind of not dialed in on all that. I have the VNS guy, which is my posting sort of launching pad on Instagram, but I don't do that much of it. I'm on LinkedIn as my name, JP Errico, and I try to be responsive to people when they reach out to me, but I'm focused on writing these papers, and I'm focused on doing the research, and I'm focused on learning new things and sharing with people. And I am going to be coming out with a book. I have one book that's already out there in this topic. It's called The Vagus Immune Connection. Easy to read. It's for the layperson. It's not too technical. And I really want to take these upcoming 20 or so papers and condense them down into something that's digestible for maybe it's not the average lay person, but for the biohacker, for the biohacker
the intermediate,
who's not a hard scientist, but is interested and wants to read about these things. And I'm going to try to frame the whole story of what actually is aging and how it is that we can avoid chronic disease and how we in the future may be able to reverse it.
Yeah. Oh, I can't wait to read it because that sounds fascinating and right down my alley. And I will be for sure putting all of the studies, at least a few of them, definitely yours that you mentioned, in the show notes so people can dive deeper. And there's another study that I wanted to put in the show notes because we've talked a little bit about epigenetic drift. And about a year ago, I came across a study that was published in Human Molecular Genetics about epigenetic drift and what its implications are. And that beautifully describes what it means and has a graph that I remember at the time I was like, this makes perfect sense. So we'll put that in the show notes. And if you guys, our listeners, have any questions, comments, please drop it below because we want to hear from you. We love hearing from you. And as always, please subscribe, like, comment, and share it with somebody who needs to hear how you can upstream address the pace of aging. And it doesn't have to take and go as far as genetic reprogramming, or at least not yet. So thank you so much, JP, for taking the time to come on. And we'll have you back on, I'm sure, sooner than later, because you have a lot going on, and I can't wait to hear more about it.
Looking forward to it. Thank you very much for having me.
Thank you so much.